Welcome to Lesson 1 of Module 1, Cells as the Basis of Life. In this video, we're going to be looking at different cell varieties, so mainly prokaryotic and eukaryotic cells. We're going to look at the organelles that they possess, and we're going to compare the two different types of cell arrangements. And the inquiry question which is focused on in this video is what distinguishes one cell from another. When we talk about cells, we're talking about the basic structural unit of all living things. And when we talk about the different types of organization, we're talking about prokaryotic or eukaryotic. When we refer to prokaryotic cells, we're talking about unicellular cells with organelles that are not membrane bound, whereas eukaryotic cells contain membrane bound organelles. When we use the word organelles, we're talking about subcellular structures that each have a specific function within the cell. There are four types of common cell structures between both prokaryotes and eukaryotic cells. We have the cell membrane, which separates the interior of the cell from the outside environment. We have cytoplasm, which is the liquid part of the cell, which contains cytosol, and that's because it contains water, ions, salts, and organic molecules. DNA, so that's the hereditary information which directs the cell's activities. And then they both also have ribosomes, which are organelles responsible for the synthesis of proteins. Here we have just a picture of the plant cell, an animal cell, and then on the far right we have a bacterial cell with some of the most common organelles, and not all structures are visible in these diagrams. When we talk about prokaryotic cells, they are generally smaller and less complex than eukaryotic cells, and being much smaller allows them to have a large surface area relative to their volume, which allows for efficient, so very fast uptake and release of materials from their environment or to their environment. Their organelles, again, not membrane bound, so they float around inside that cellular membrane. And prokaryotic cells are divided into two domains, both bacteria and archaea. When we talk about bacteria, and archaea, we're talking about microscopic single-celled organisms. Bacterial cells have a diverse metabolic systems depending on the type of bacteria. Some obtain their energy from sunlight, and some obtain their energy from reducing inorganic compounds. Their cell walls are made up of peptidoglycan, and they can either be gram-positive or gram-negative depending on the thickness of the peptidoglycan layer, which depends on the type of bacteria and some of the characteristics that they then have. When we talk about archaea, we're talking about extremophiles, which live in extreme conditions, so such as high salt, high temperatures. There's also some archaea which produce or obtain their energy from methanogenesis, which is the breaking down of methane. Their cell membrane is composed mainly of lipids, which allows that structure to remain fluid and selectively permeable over that wide range of temperatures in which they might reside. So here we just have some diagrams. We have bacteria and archaea on the left, and then we have another type of bacteria called cyanobacteria on the right. When we're talking about eukaryotic cells, we know that they're much larger and more complex, and because of this, they can either be unicellular or multicellular, and they can either produce asexually or sexually. Eukaryotic cells have a cell membrane that surrounds the cell, and they have internal membranes which form specialized compartments, which allows for these membrane-bound structures, which we refer to as organelles. Some of the benefits of compartmentalization, which is that it allows the enzymes and the reactants for particular cellular functions to be close together in high concentrations. And it allows that because they're in those small pockets at the right concentrations, it allows many different environments to be occurring at the same time within the same cell. Therefore, this makes the cell less vulnerable to changes in its external environment. So here we just have examples of eukaryotes. So we've got some fungi, protists, and we also have plant and animal cells. We're just going to go quickly through some of the different organelles which might be present in the different types of cells. So we have the nucleus, which is surrounded by a nuclear membrane which contains the genetic information of the cell. And within eukaryotes, they're in linear chromosomes, and in prokaryotes, they're usually circular structures. We have ribosomes, which are composed of proteins, and they're the sites of protein synthesis. And they translate the sequence of amino acids specified by mRNA into proteins. We then have the endoplasmic reticulum, which is a network of little sacs and tubules, and this can either be rough or smooth depending on whether they have ribosomes attached or not, and they're involved in the synthesis and the production and the export of proteins. We then have the Golgi apparatus, which is a stack of flattened membrane sacs, and these little sacs can form little vesicles which transport proteins either within the cell or out of the cell. We then have lysosomes, which again are little vesicles, and these lysosomes actually digest unwanted matter from the cell, and they can either bring these little packages into the cell and transport them around the cell, or they can transport them out of the cell. We then have the mitochondria, which contains DNA, and this plays an important role in cellular respiration within eukaryotes. We then also have the chloroplasts, which also possess DNA, which is involved in photosynthesis within plants, and these trap light energy for energy production. We then have vacuoles, which are membrane-bound spaces which can store enzymes and other materials. In animals, these vacuoles are small and usually temporary, and then in plant cells, these vacuoles, there's usually just one large permanent structure, and this is really important for the structural support of plants. We then also have plastids, which are involved in the synthesis and storage of chemical compounds, and these might be referred to as either chloroplasts, luteoplasts, or chromoplasts. We then have a cell wall, which is present in plant cells, which surrounds the cell membrane and provides structural support. It also prevents expansion, but it allows water and substances to pass freely through it. We then have the cytoskeleton, which are microtubules and filaments, which supports the structure of the cell and allows it to move, and it transports the organelles within the cell. We then have centrioles, which are involved in cell division. And then finally, we have cilia and flagella, which are little hair-like structures which are involved in the movement of the cell or things around the cell. Our final slide for this video is where we're going to compare both prokaryotes and eukaryotes. So the first one is we compare the membrane-bound organelles, and we know that prokaryotes don't have these, whereas we see them in eukaryotes. The nucleus and DNA within prokaryotic cells, they have a single circular chromosome and plasmids, whereas eukaryotic cells have linear chromosomes that are contained within the nucleus. Cell walls of prokaryotic cells have peptidoglycan, where the cell walls and membranes of eukaryotes have carbohydrates. And then we've also heard many times that the eukaryotic cells are usually 10 times larger than most prokaryotic cells. Thank you for watching lesson 1. Make sure you tune in for lesson 2.